Good afternoon, and welcome to another edition of the CannaList Conversations. I am your host, Patrick Daugherty, and I have the pleasure of being joined today by Dr. Ethan Russo of CReDO Science and Chinga Jungson of Canurta. Welcome. How are both of you today?
And thank you.
Great, thanks.
Great. So, Dr. Russo, you've been working in this field for a while. You have your company, CReDO Science. Can you give us a little bit of your background and what you're working on?
Sure. Well, I'm a neurologist by training, but I've been involved in medicinal plant research for 30 years. Some of that was while I was still in practice. This was prompted by my feeling that I was giving increasingly toxic drugs to my patients with less and less benefit, and my desire to seek alternative pathways, hopefully with plant-based medicines that might provide similar or better results with a greater safety profile. So I really started out doing ethnobotanical work with a particular focus on the Amazon rainforest, which culminated in 1995 with a 2.5-month sabbatical in the rainforest in Peru with the Machiginga tribe. When I came back, I knew that I wanted to transition into a full-time research capability, but it took me some time. But by then it was 1996, and I quickly became embroiled in what we'll call the cannabis controversy. That was the same year that Prop 215 was beginning in California. But very rapidly, I became enamored with the field. Cannabis is a very complex topic, everything from its growth and habits, its biochemistry, therapeutic usage and history. It was sort of a bottomless pit of information that I delved into with both feet. This culminated in trying to do research in the US through the FDA, but I was largely stymied by the roadblocks to research, which for the most part are still in place. But I began writing and publishing, and came to the notice of GW Pharmaceuticals, the company that has developed what to this point are the only two cannabis-based pharmaceuticals in the world. So I was an advisor to them starting in 1998, the year that the company began, and had a full-time position with them as senior medical advisor starting in 2003 for the next 11 years. So I was heavily involved with the phase 1 to 3 clinical trials of Sativex and Epidiolex, as well as being a liaison to basic scientists around the world that were supported by GW in investigating cannabinoids, particularly most of the early work on cannabidiol was done under those conditions. Subsequently, at the end of 2014, I transitioned into a couple of private companies, and then 2 years ago, decided that I really needed to do this on my own, so began CReDO Science with my business partner, Nisha Whiteley. In the course of this, we had many ideas that had been percolating that I had not been able to develop in my prior business relationships. So we began by trying to get intellectual property protection in a number of areas. One of those of interest related to canflavins. So we need to explain what those are. If you don't have an intervening question, I'll just continue.
No, go ahead.
This has got a really interesting history, and it hits me personally because the person who discovered this, somebody I know but didn't know at the time. So Marilyn Barrett was an American scientist who was studying pharmacognosy, the medicinal properties of plants in England in the 80s. They were looking at anti-inflammatory substances in cannabis, and she discovered two closely related molecules called canflavin A and B. And they turned out to be some 30 times more powerful than aspirin as anti-inflammatories, and twice that of hydrocortisone. But not a lot was done on this. Truth be told, THC really sucks up all the bandwidth, or did in the past, in relation to cannabis research. Slowly, cannabidiol has taken more center stage, but what has been ignored is some of the hundreds of other compounds in the plant that have medicinal applications. So not much was done with canflavins for some time. I subsequently met Marilyn in the rainforest in 1994. She was working for a company called GW Pharmaceuticals, which was a company devoted to trying to do pharmaceutical development on medicinal plants. So this was very much up my alley, and I had an association with them for a short time. But I didn't know that she was the M. Barrett that was discoverer of canflavin for some years. Fast forward to 2014. Giovanni Appendino is a medicinal chemist in Italy. He and his team, including Oliver Wertz, W-E-R-Z, collaborated on a study in 2014 where they looked at canflavins, and they had isolated these in an interesting way by growing hemp sprouts. So when we have a seed of cannabis, if it's been properly washed, it has no cannabinoids on it. And it usually won't develop any cannabinoids until it's further along in the growth process. And they discovered something very interesting, which was that there was a reciprocal relationship between the canflavin count and the cannabinoid concentration. In other words, if you had a chemovar, chemical variety of cannabis, that produced little or no cannabinoids, it tended to produce more canflavins. It was like the plant had the wisdom to say, we're missing something on this side, and we need protective compounds from somewhere else. Generally, canflavins are found in the developing plant and in the leaves. They're really not in the glandular trichomes, which are where the cannabinoids and terpenoids are produced. So these are sort of complementary compounds that the plant produces for its own ecological reasons. Certainly, they're not there to get us high or to be medicine for humans, much as we'd like to anthropomorphize the situation. But in their study, Wertz et al. Looked at canflavins and established some of the mechanisms by which these were effective anti-inflammatory agents. And so what they found was they worked on the two major inflammatory pathways that affect people and animals. And those are called the lipoxygenase and the prostaglandin pathways. Now, this is quite in contrast to the standard anti-inflammatories. On the one hand, we've got steroids. These are very important under conditions of serious illness, but they have very marked side effects, including weight gain, ulcers, susceptibility to infection, thinning of bones. So they're not something that people can take chronically without a lot of issues. So these were supplemented in medicinal research by the non-steroidal anti-inflammatories. The most famous one being aspirin, but subsequently naproxen, ibuprofen, the common drugs that people use to treat pain or inflammation. Now, the problem there is they're also subject to very serious side effects, even over-the-counter preparations, because these can lead to ulcers, gastrointestinal bleeding, and also have a role in making heart attacks and strokes a little more likely if people are taking them regularly. In contrast, the canflavins have none of these adverse event potential. So they do not inhibit an enzyme called COX, cyclooxygenase, in the same way, and aren't subject to producing the same kind of side effects. So the idea being, if we have a source of canflavins that doesn't have these liabilities, it might help with any number of diseases that have at their basis inflammation. And that's so many things. It runs from infectious disease to cancer to degenerative disease like Alzheimer's disease. So the potential there is enormous. The issue then became, how do you get this stuff? Normally, canflavins are produced in the plant in small amounts. Now, as I mentioned, certain plants have a genetic tendency to produce more. So selective breeding is one way. The other way would be extracting from biomass. In other words, let's say as a byproduct of the medicinal cannabis industry or the recreational industry, that there was a lot of leftover biomass. Is there a way to get canflavins out of that? And that's part of the work that our colleagues at Guelph, the university in Canada, accomplished. But they went one step further. They worked out the enzymes involved in the production of canflavins and figured out a way to make these biosynthetically. So let's just say, for the sake of discussion, that we had a condition where we needed big doses of canflavins. Under those conditions, it might be most practical to make these biosynthetically and supply the kind of doses that might be necessary to treat cancer, for example. In the course
it
of
would have the
this,
same efficacy as the biosynthetic would have the same efficacy?
well, the molecules would be the same. So to the body, it would be the same thing. As long as the three-dimensional structure of the molecule is the same, it could work just as well. Now, I will state that normally I prefer complex mixtures, extracts from plants, but there's certainly certain contexts in which you need bigger amounts, and biosynthesis of a single molecule, a single compound, may be the most expedient way to go. It also quite clearly is the pathway that is more recognized by the Food and Drug Administration and other regulatory bodies. It's what they're used to. They've had a little more trouble understanding botanical pharmaceuticals, where there isn't a single active compound, but rather a mixture that produces a therapeutic effect. So yeah, a little more complex.
And the botanical approach, they feel there's less control, correct?
Well, that's one of the pitfalls. Can be achieved. For example, Sativex, which is an approved drug in 30 countries, not currently in the US. This is a complex mixture. It is a plant extract. It was recognized as a unitary formulation, and it did get through what's called chemistry, manufacturing, and control at the Food and Drug Administration, meaning that they were able to show consistency over time, batch to batch, in the various components within tight parameters, so that it would meet the stringent requirements of quality and consistency to be a pharmaceutical drug.
And then, so where does this stand today? You've been working on this at CReDO. How far along are you? Are we ready for evidence-based trials? Where are we to?
Yeah, close. One of the things that's nice about our collaboration with Canurta is that very stepwise approach is planned. We're starting out with sprouted hemp seed. People may be aware of hemp hearts and similar products that are available now on the market. And these are often grown in Canada and imported in the US, and there's no cannabinoid content, so really no controversy. Sprouted hemp seed will have additional nutritional benefits. Also, we hope that we'll be able to produce a product that has greater amounts of canflavins in them, so that this would be available as an ingredient for granola or part of an ingredient in baking. There would be any number of possibilities. So that's the first step. The second would be more refined preparations that could be used as supplements, for example, in capsule form or combined with other botanicals, again, to join the many supplements on the market, many of which are plant-based already. And then the third echelon of activity would be the goal of pharmaceutical preparation. Obviously, that is a much longer and more expensive proposition. It often involves hundreds of millions of dollars to get
it's usually a B. Usually comes with a B price tag, right?
Yeah, again, we like to say 800 million to 1.2 billion. Now there are
and 8 to 10 years of,
yeah, that certainly is the case. So that one's down the road, but you have to begin somewhere short of that. And that involves things like toxicology studies, where we show that high doses of the compound don't have any deleterious effect on the body. And unfortunately, using test animals, the current requirements are that new drugs be examined in this way in 2 species, usually rat and dog. Additionally, there'll be other basic science approaches to see what types of disorders or diseases are really amenable to treatment with these compounds. And again, animal work can be done as a prelude to human clinical trials. And those go through 3 phases. Phase 1 is what's called pharmacokinetic pharmacodynamic studies, in which you take normal volunteers and give them a certain amount of the preparation, measure the amounts in the blood, and monitor for any side effects. Phase 2 involves smaller studies of patients with a particular disorder using the drug to demonstrate safety and efficacy. And phase 3 is larger numbers of people with a disorder, again, safety and efficacy. And the third measure is, again, consistency based on laboratory analysis. But again, this is a path that's been taken before with botanicals, let alone isolated compounds. It's been done properly and before, and can be done again.
So the initial approach would be a supplement, whereas you won't be making any claims with regard to inflammatory.
Sure. Well, in the U.S., you can make structure activity claims. So these are vague notions of how an agent may work. So you could say, for example, supports health of the gastrointestinal system. What you could not say is effectively treats Crohn's disease. Right. So those are the distinctions.
And so what made you pursue a partnership? Can you tell us about that?
Well, it was obvious that we had common interests in these substances. Originally, we had a more modest goal in this of producing canned flavins from sprouts and putting them, say, into nutrition bars, things of this sort. But at the time, we didn't have someone to co-develop this. CReDO Science is not a manufacturer or marketer, so we were going to need that anyway. And in our early discussions with Canurta, it became apparent that they really had the team in place to show the efficacy of these compounds and develop it down these various pathways. So, it's much better to work as part of a team than to try and go it separately, if you will. So beyond that, we liked the people very much, and it was a very easy decision to develop a partnership.
So Canurta will take on the role of bringing it to market and to market and distribute, correct?
Right, you bet. Now, along the way, who knows what happens? Typically, somewhere after phase 2, if a company has shown that they have the safety and efficacy up to that point, often they will develop a partnership with a bigger pharmaceutical company, because
It's expensive.
the later stages are difficult. It really takes a very big company to successfully market a drug, and getting through phase 3 is a much bigger step than anything preceding that. And usually that requires big companies' involvement. So that would be a possibility down the road. But what the trend has been of late is that most of the big pharmaceutical companies have very little internal research and development. They leave it to smaller companies, innovative companies like Canurta, to do the initial work, and then they just partner up with them after they've done the initial heavy lifting.
Yeah, it's nice for them to let you take the risk, and then once you've proven it, then they're willing to jump on board.
Yeah, that's the case.
I can say that. You don't have to say that. I'll say it. So,
Well,
but
I don't disagree, certainly.
So what do you where do you go from here? You're
Well,
moving it along. What's the next steps?
We're extremely excited. Initially, we had some ideas about where these would fit into things. I mean, the obvious things would be aches and pains and arthritis, but there's evidence of much bigger things possible. Similar molecules have proven to be efficacious in treating various types of cancer. It's just, there may be a role in degenerative diseases like Alzheimer's disease. These have been very thorny problems for decades that have still resisted best efforts in medical research to deal with them effectively. We have what I really presume to be a tremendous safety factor here. And given that as a place to start, if you have something that's safe, that has the ability to contribute to treating or even curing some of these more intractable conditions, that's always exciting.
And in your opinion, is Western medicine there yet? Do they still look at cannabis as an alternative treatment, but not ready to embrace it, or are they starting to move towards embracing cannabis and starting, to prescribe it once it's been through the trial process?
Yeah, it's been a slow evolution. I mean, when I started this kind of work 30 years ago, my colleagues thought I was nuts. But when they saw the kinds of results I was getting with their patients and using natural products, including cannabis, they came around. Unfortunately, with cannabis, what it often takes is for a close family member, the person, him or herself, to have some bad condition that's benefited from cannabis-based treatment to realize the utility. As I alluded to previously, there's still considerable roadblocks to research in the US. But here we're talking about something different. When people think about medicine and cannabis, they're thinking about THC and maybe CBD. We're in a different ballgame here. These would be what are considered minor molecules in cannabis. I can tell you that canflavins, there's no indication that they'll be intoxicating or have any drug abuse liability. So this is something else. This shouldn't be controversial. It may be controversial to some people, the idea that any plant-based compound is going to be as good or better than what the pharmaceutical industry can produce from scratch. But I can argue that one until the proverbial cows come home.
And it seems that the pharmaceutical industry is embracing this, right? Because they're recognizing the potential.
Sure, it remains the case that at least 25% of current pharmaceutical medicines are either directly come from plants or have been based on molecules that came from plants that they've dickered with to make them their own, so to speak. So before synthetic chemistry in the last 75 years, medicine was from plants.
Hmm.
And our species has managed to survive all this time. So there had to be some good there.
Yeah, but there weren't the giant pharmaceutical companies back then that there are today that have a vested interest in, other products, right?
Sure. Well, that you really have put your finger on part of the problem. It is the preference generally of pharmaceutical companies to synthesize their own molecule to which they have intellectual property rights. They can patent it. One of the misconceptions is that you can't get any intellectual property protection for the kind of development that we're discussing. You can, but it has to be focused. You can't patent the cannabis sativa as a species. However, you can patent a preparation or an isolation technique, or get a use patent that demonstrates efficacy in a particular disorder. So there are financial incentives that are available even for botanical medicine, and that's certainly the case. Again, there's a path that's been laid already, and it's one that we hope to follow to the good of all.
And then the go-to-market will be initially supplement route, right?
Sure, nutrition first, then likely supplements, and hopefully eventually pharmaceutical preparations.
And is the plan to go through Canada to start in the US? What is the initial sort of go-to-market?
Well, I'm going to let you go through that more with Akeem, the CEO, in subsequent conversations. But normally most of the activity is going to begin in Canada, and certainly this will be done in conjunction with Health Canada. But there's always a viewpoint towards the FDA, which of course is the big enchilada in terms of the medical market. But we would hope to have products that would be utilized worldwide ultimately.
And are you finding, there's a lot of activity going on in this space, particularly from the medicinal side. Is this a crowded landscape, or are you out on your own and sort of found something unique?
There are companies that are interested in synthetic analogs of the molecules that we're discussing. Again, this is my personal bias, but based on informed opinion, I think that they've got a longer pathway. We, again, are starting with material that is going to be nutritional. We'll have the opportunity to test this and demonstrate safety, etc. I think that we can produce safe and effective medicines in a much shorter time frame than any company that would be looking at a semi-synthetic or wholly synthetic preparation.
Great. Well, Dr. Russo, we very much appreciate your time today. Shenga, thanks you for joining us as well. Our intent is to have a subsequent conversation with the founder of Canurta, Akeem Gardner, and that will come along as well. So look forward to that. Again, thank you both for your time, and we look forward to great things coming from both partnerships. Thank you.
Thank you.